Hello and welcome. Today we'll be discussing about the histology of circulatory system. So in this we'll be discussing about the histology of the blood vessels, lymphatics, including the heart. So let's start. So as you know that the circulatory system basically consisting of two subsystems in the body. One is the cardiovascular system and one is the lymphatic system. The cardiovascular system basically constitutes the heart, arteries, veins, and the capillaries. Are the lymphatic system as the lymphatic vessels, lymphatic capillaries, lymphoid organs, and the lymphoid ducts. So in the cardiovascular system, the central pumping organ is the heart, and the conveying channel are the blood vessels, and the medium that is flowing those through these blood vessels will be the blood. So cardiovascular system basically contains heart, arteries, veins, capillaries, sinusoids, and the cavernous tissues, and we'll be looking at the histology of all of the things in this lecture. And if you know that blood vessels basically two systems we have discussed it, that two systems they are present in the body. One is the arterial system, one is the venous system. So arterial system basically that transports the blood away from the heart, and venous the blood is transported towards the heart. The arterial system contains the arteries, arterioles, capillaries, while in the venous system you have the venules and the veins. So the basic structure of any blood vessel, be it artery or the be it vein, will be having these three layers. One is what we call as tunics or the coats, and if you just name these three coats, one will be the tunica intima, that is the innermost lining of any blood vessel that is present in the body. Then tunica media is the middle one, and tunica adventitia is the outer one. The difference between the arteries, veins, or venules, and the capillaries, or any type of the blood vessel, or any type of the vessel that is present in the body, will be in these layers. So if you look out, the first is the tunica intima, the innermost layer. So in the tunica intima, that is the innermost layer, we can find the lining epithelium, that will be the endothelium of any type of vessel. And the lining epithelium, endothelium, basically the simple squamous type of epithelium. Then this epithelium will be followed by a thin basement membrane. And under this basement membrane, you have a small amount of the connective tissue, what we call as subendothelial connective tissue. And then this subendothelial connective tissue will be supported by one of the elastic lamina, what we call as internal elastic lamina. And this internal elastic lamina basically is fenestrated, or there are the openings in this lamina. And the fenestration basically provides the passage for the nutrients. And this internal elastic lamina basically separates it from the other layer of the vessel, that is the tunica media. So this is the basic structure for any type of vessel. The tunica intima should have these layers, and the difference will lie in these layers also between the different type of the vessels. So tunica media, what we can find in the tunica media, so maximum differences lies in the tunica media between the vessels, and one can find the concentric layers of smooth muscles in this layer. You can see the smooth muscle spindle shaped cells here. Those are the smooth muscle cells. Plus, besides the smooth muscles, you have the elastic fibers also. Then you have the few collagen and reticular fibers, and this tunica media, at one side it will be separated from the intima by the internal elastic lamina, but on the other side it will be separated from the tunica externa by what you call as external elastic lamina. Then this tunica media, I told you that the difference will be here in between the vessels. So it will be thicker, very thicker in case of the arteries, and then the veins. So veins will have one of the difference between the artery and vein is that tunica media will be thicker in arteries as compared to that of the veins. And if you go for the smooth muscle contribution or the elastic fiber presence in this layer, you will have more smooth muscle and elastic fibers in the arteries as compared to that of the veins in the same size in the tunica media. Tunica adventitia, that is because this will be continuous with that of other organs, and it will be with the stroma of other organs, and excessive stretching of any type of blood vessel will be prevented by this tunica adventitia. And the maximum contribution of this tunica adventitia will be of the collagen fibers, and basically this is a connective tissue. So here more of the collagen fibers will be present and few elastic fibers. And if you look out the fiber predisposition in different type of the layers, in case of tunica intima and tunica adventitia, you have the maximum of collagen fibers. Those run longitudinally, and in case of tunica media, you will have more of elastic fibers, and those elastic fibers basically run in the circular fashion. To talk about the endothelium, the innermost lining, what I told you that it will be made up of the simple squamous type of epithelium, and it will have the cytoplasm, endoplasm, reticulum, mitochondria, and other things in these endothelial cells. So coming to the first system, that is our arterial system. So arterial or the arteries basically are the thick walled blood vessels that carry the blood away from the heart, and they divide continuously, and you will find a branch of tree like that of when they divide. And when you go from heart to when you go when these arteries just move away from the heart, usually the rate of blood flow generally decreases, plus the lumen will also decrease when this artery will go away from the heart. So you have the different types of arterial system in the body. The one is elastic arteries; they are the large arteries, and in this type of arteries, you have the more elastic fiber in the tunica media. So this is one of the differences in the arteries also. So elastic artery, you have more elastic fiber in the tunica media and less of the smooth muscle. Then muscular artery, you have more of the smooth muscles in the tunica media and the less of the elastic fibers in tunica media. Then arterioles, you have 1 to 2 layer of smooth muscle in the tunica media. Then capillaries, you have only tunica intima, and there will be the underlying basal lamina. That means internal elastic lamina will be absent in case of capillaries. So the elastic arteries, also known as the conducting arteries, so they conduct the blood away from the heart and just move this blood towards the smaller arteries. And the other name of these elastic arteries are the conducting arteries. And if you look out the histology of these elastic arteries, so if you see the tunica intima, so you will have the epithelium as endothelium as a simple squamous type of epithelium. Then you have subendothelial connective tissue also present. Then there will be the presence of this internal elastic lamina, and then this internal elastic lamina will not be clearly distinguished because the next layer, that is the tunica media, will be also having the enormous amount of the elastic fibers, so that this internal elastic lamina will get merged with that of other elastic fibers of the tunica media. So one cannot see clearly the internal elastic lamina of tunica intima because of the underlying tunica media is also having a lot of elastic fibers. So tunica media, you have a large amount of the elastic fibers. You can see in this histological picture, a lot of the elastic fibers and a few smooth cells are present. And because of this, you cannot differentiate the intima and the media. So the other side of the tunica media, you should have the external elastic lamina, and this also you cannot differentiate because of the abundance of the internal abundance of the elastic fibers in the tunica media. So tunica adventitia will be very thin in the elastic arteries, and you will find some of the elastic and the collagen fibers in this tunica adventitia, and that runs longitudinally. Plus, in this adventitia, you will have the small vessels that are supplying towards the large vessels. We call them as a vasa vasorum. So one can find the vasa vasorum in the tunica adventitia. So these are some of the features of elastic artery that we have already discussed. So elastic artery, basically, if you see the picture of elastic artery under the microscope, under the 4x, you can see this internal elastic lamina, this tunica media, and tunica adventitia. So these are some of the examples in the body where you can find the elastic artery. You have the aorta, brachiocephalic, carotid artery. So these are the big arteries which are the elastic in the nature. So these are some of the examples of H&E stained, and you can find on my right, you can find so many of elastic fibers here. So this elastic artery here also, so many elastic fibers, wavy appearance of the fibers, those are elastic fibers here also. You can find this is the trichrome stain. You can find here so much of elastic fibers there. Then the muscular artery. So muscular artery are also known as the distributing arteries. They are the smaller than that of, or they are the medium-sized arteries, smaller than that of the elastic arteries. And if you see the differences in the muscular artery in the tunica intima first, so you will have in these type of arteries, there will be the endothelial lining, there will be subendothelial connective tissue, there will be internal elastic lamina, and this will be well distinguished because the second layer, that is tunica media, will be having more of the smooth muscle cells and less of this elastic fibers. Because of that, there will be a contrast difference or contrasting feature that internal elastic lamina can be easily seen. Tunica media. In the tunica media, more of the smooth muscle cells will be present and arranged in the circularly, and there will be some few collagen and elastic fibers. And external elastic lamina can also be seen very well because of presence of the smooth muscle cells in the tunica media. Tunica adventitia, it will be thicker than that of elastic artery. So tunica adventitia is thicker in muscular artery as compared to that of elastic arteries. And vasa vasorum can also be seen in the tunica adventitia of the muscular arteries. These are some of the examples of the muscular artery, femoral, popliteal, radial, ulnar, and external carotid artery branch. So this is muscular artery. You can find, visualize this internal elastic lamina very well. Then this is the tunica media. More of here will be the smooth muscle cells. So you cannot appreciate the elastic fiber here. Then this is tunica adventitia. Here you can see this is an enlarged view of the muscular artery. So this is wave appearance of the tunica internal elastic lamina of tunica intima. Here also you can see that. So this is the trichrome staining of the muscular artery. So you can, this is the lumen of artery, muscular artery. This is the internal elastic lamina. This is the tunica media. So one cannot appreciate the elastic fibers there. Then this is adventitia, and this purplish is again external elastic lamina. So these two laminas, external internal elastic and external elastic lamina, are quite appreciatively seen in the muscular artery because of the presence of the much of the spindle muscle or the smooth muscle fibers in the tunica media. So this is some of the differences between elastic artery and muscular artery that we have already uh, discussed. These are some of the pictures of between the muscular and elastic artery. These are the histological differences between the two. So coming to the arteriole, so these are the small arteries with a diameter less than 100 microns, large arterioles and terminal arterioles, and basically they control the blood flow, and major determinants of the systemic blood pressures are these arterioles. So basic structure of arteriole, no subendothelial connective tissue in the tunica intima. Tunica intima will be very thin. Internal elastic may be absent or very thin in the arteriole. Tunica media is also very thin, and there will be no external elastic lamina. And tunica adventitia will be very poorly developed, thin or very poorly developed in the arteriole. So these are the arterioles you can see here. Tunica media is not that much of developed, and external lamina is not that much of seen here. So terminal arterioles, basically the diameter will be less than 50 microns, and smallest among the terminal arteriole will be 12 microns, and they will be having the one only a thin layer of muscles in their walls. And when they branch off these terminal arterioles, they are called as meta-arterioles. So meta-arterioles are the smallest of the arterioles, and they have the discontinuous layer of the smooth muscle layer. Then this meta-arterioles will finally terminate into the capillaries. You can see that capillaries have 2-3 types that we'll be discussing in the coming slides. So the capillaries, basically they are the smallest blood vessels. The terminal arterioles basically just end into the capillaries, and the average diameter will be 8 microns. And they are the very thin walled, and they are basically the site of exchange of gases, nutrients, and metabolic wastes. So basic structure of any type of capillary, you will have a single layer of endothelium cell lining. Then you have the basal lamina. Then you have the contractile cell outside of the basal lamina, that's called the pericyte. And this capillary basically has only tunica intima, so media and adventitia will be absent. So there will be no smooth muscle because the tunica media will be absent. That's why it has a pericyte as a contractile unit. So types of capillaries in the body, you have the continuous type of capillaries, you have the fenestrated type of capillaries, and you have the sinusoidal type of the capillaries. So continuous capillaries, you have the basement membrane and you have the endothelial linings. Those are continuous. There is no fenestrations here. So both the layers, basement membrane and including the endothelial cell lining will be continuous. Fenestrated, so basement membrane will be the continuous, but there will be the fenestrations or the openings in the endothelial linings. So example are kidney, small intestine, endocrine glands. You can find the fenestrated type of capillaries there. And sinusoidal, there will be the fenestration in both the basement membrane will be discontinuous. That means fenestrations will be there, and there will be the fenestrations in or the pores in the this endothelial lining also. You can find these in the liver, bone marrow, spleen. You have the example of the liver as a sinusoidal organ. So this is the picture of histological picture of the liver. You have the central vein, hepatocytes, and you can all these white spaces are basically the sinusoids. So these are the H&E staining, and this black and white is basically the electron microscopic picture. Here also you can find the capillaries in the adipose tissue, the white or adipose tissue, because the white adipose tissue will not get stained by H&E. So this is some of the difference between the capillary and the sinusoid. So coming to the venous system basically the thin walled. As we already discussed that there is a difference between the vein and the artery in the three layers, and usually they will carry the blood from the capillaries towards the heart, and these large veins basically are formed by the smaller veins, like there are the tributaries of a river. And besides these veins, they will be supplied by the valves which will prevent the backflow of the blood. That means there will be the unidirectional flow of the blood towards the heart. Three types of the veins you can find: large veins, medium-sized veins, and the venules. So layers will be same as tunica intima, media, adventitia, like that of arteries. The difference will be regarding the presence, absence, and the thickness of the different things that are present in these three layers: intima, media, and adventitia. So one can find the difference in the tunica media is very thin in case or poorly developed in case of the vein. You can find this the below diagram. You can have the tunica intima, but the media is very thin. Above this picture is of arteries. You can see the middle layer is that pinkish layer is very thick. So this vein contains more of the collagen fibers, less of the elastic tissue. And if you see the lumen of artery and the lumen of the vein, usually the lumen of the vein is usually collapsed after the death of any human being or animal. Whereas in case of artery, the lumen will be as such what is present in the live condition. Tunica intima and adventitia are more prominent in vein. Tunica adventitia will be thicker than the media. Internal and external elastic lamina are difficult to distinguish in the vein. So this is the small artery and the small vein. You can see that the level of the thickness between the different walls in the vein and artery. But you can see the wall of thickness to the lumen diameter. It will be more in case of the vein as compared to that of artery. That means the lumen will be more in case of vein as compared to the artery, and that is because of the wall thickness will be more in the artery, which will because of that there will be the less of lumen as compared to that of the vein. So muscular artery and the vein, you can see the level of muscular uh, tunica media here, the level of thickness in the muscular artery and the level of thickness in case of the vein. So large veins, if you see the histological picture of the large veins, you have endothelium and subendothelial connective tissue in tunica intima. So tunica intima will be very developed in the large veins. Tunica media will be very thin because of less smooth muscle and elastic fibers will be present in the tunica media. So tunica adventitia will be very well developed, and tunica adventitia there will be the small blood vessels that was called the vasa vasorum. So internal and external elastic lamina are poorly developed in case of the veins as that of the large vein also. So examples of large veins, you have SVC, superior vena cava, and inferior vena cava. So here again, tunica adventitia is highly developed in case of large veins. So medium-sized veins, you have the size of the vein is as 1 to 10 millimeters in the diameter. Examples are femoral vein and the superior mesenteric veins. And tunica intima is very thin, tunica media is less muscular, and tunica adventitia will be well developed and but will be thicker than that of the tunica media. So these are the veins. The first thing you can see the collapsed nature of the lumen. This is H&E stained, and this is that you can find as a multicolor. This is basically a trichrome stain. You can see the lumen is basically collapsed. Then if you see here, the adventitia is more as compared to that of the media. So adventitia is highly developed or well developed in case of veins. So this is the difference between the medium-sized vein and medium-sized artery. So artery and vein, again the difference. Venule, so like that of arteriole, you have the venule here, the smallest veins, and basically the capillaries that are the end of the meta-arterioles, they will drain and the blood into these post-capillary venules, and what to call as a venules, and these post-capillary venules then drain into large muscular venules, and finally the big vein will be formed. So wall is very thin, and the lumen is highly collapsed. Tunica intima will be having only the endothelial lining. Tunica media will be 1 to 2 layers of the smooth fibers will be present, but the tunica adventitia will be as thicker, because the feature of any venule is that tunica adventitia is generally thicker as compared to that of a tunica media. You can see this is the venule, this is the arteriole, and this is the connective tissue, this is the duct. So one of the feature of what I told you in the initial is the valves in the vein. So these valves you can see sometimes under the H&E staining. So this is H&E stained. You can see that they will come to the lumen. So these are the folds of the tunica intima forming the valves. So because of these valves, there will be the unidirectional flow of the blood towards the heart. So this is a diagrammatic representation of the valves in the vein. You can see here. So when they close here, so the unidirectional flow of the blood is prevented. Again here, we have the cusps type of valves here. So these are the changes that happen to the different type of vessels due to age. You can just consult any type of book, or you can see there are many sources to see the change in the arteries. These are again the change in the arteries. So if you see the histological picture of the heart, and basically the heart has three linings: one is endocardium, one is myocardium, and one is epicardium. So endocardium is the innermost endothelial lining, like that of any type of blood vessel. Here also the internal lining of the heart is endocardium, what we call as endothelium, plus subendothelial connective tissue will be there, like that tunica intima. Myocardium is the main part, that is the cardiac muscle. Then epicardium is the covering. You have the pericardium as covering of the heart. So these are some of the features, histological features of endocardium: simple squamous type of epithelium; myocardium, thickest layer of cardiac muscles; epicardium is a loose connective tissue. So this is the diagrammatic representation from interior to outside. So this is endocardium, the thicker is the myocardium. Then there will be the pericardial cavity, because the pericardium itself is divided into fibrous pericardium and the serous pericardium. And the serous has two layers. One is called the parietal layer, one is called the visceral layer. The visceral layer will be attached towards the myocardium. The parietal layer will be attached towards the fibrous pericardium. Between them there is a cavity, what we call as pericardial cavity, and this pericardial cavity will be filled with that of the pericardial fluid, or the liquor pericardi, and that is usually for the movement of the heart, or usually for the prevention of the friction by the movement of the heart. So this is the histological picture of the heart. You can see here pericardium, pericardial cavity, coronary artery, endocardium, cardiac skeleton. So in this picture you can see that epicardium is there, that is the inner outermost covering fibrous tissue. Then you can see here this is the muscle part, what we call the myocardium. Then there is the endocardium. Here again also we can find this is the endocardium, then myocardium. These are the fat cells, and this is the marrow, and this is the connective tissue that you find in case of the heart. So heart valve is also, if you see that basic picture will be same that that of the heart. So the basic thing what you find in the cardiac muscle is these intercalated discs, and there will be the striations, but not that of the skeletal muscle striations. You can find small striations in case of cardiac muscle also, but the basic structure what you find is the intercalated discs present between. So this is the diagrammatic representation. On the right side, you can find that the arrow here. These are intercalated discs. So besides this, you can find the Purkinje cells also in case of the heart. So the lymphatic vessels that drain the excessive fluid from the tissues. So you have the other system in the body as vessels, what called the lymphatic vessels. So the tissue fluid that is there outside the cells will be taken by the lymphatic vessels. Then they pour this tissue into the blood, and that goes towards the heart. So in case of the lymphatic vessel histology, basically the lymphatic vessel histology, again, if you see the walls of the lymphatic vessels, that is much thinner than that of the vein also. So only you can find some of the features like tunica intima, then some part of tunica media, and a lesser part of tunica adventitia in case of lymphatic vessel. So this was all about the histology of the cardio, this circulatory system. And hopefully, if you have any question, you can ask. Thank you.